So we're going to start working on Punnett squares. And I find it's always best, you know, the pea plants are cool, you know, they're purple, they're white, they're tall, you know, all that jazz. Y'all can do the boring stuff for homework. Right? Let's do something that means something to us in class, right? Let's actually have a discussion. Let's use our brains. Let's figure stuff out. So I like doing the blood typing because, again, it's personal, right? And you can form this connection with it, and it can make sense to you. So the basics of, does anybody not know the basics of Punnett squares? Because I don't have a problem going over it, but if I can skip that, then we spend more time on the fun stuff.
Can you do it like really fast?
I can do that too. I can do that too. I can do that too. Okay, so let's first start with defining some stuff for blood typing. So I need just a text box just to write some notes for us. So let's insert that. So for the ABO blood group, right, you either, and when we say you're a phenotype, that's what's expressed, right? So there's 4 different choices for blood type in that group of antigens. So we know these, right? You could be if I could type would be good. Type A, right? Or you could be B, or what? AB. And the last one? O. Okay, so yeah, Erica, an O, not a 0. Okay, so that's your phenotype, right? That's what's expressed, that's what's on the surface of your red blood cells. And how many of you guys actually know your blood type? Okay, cool. How many of you know either your children or your parents' blood type? Sweet, we got a whole bunch of Punnets we can do. Yay! All right, cool. Okay, at least a couple examples. Okay, so you get that information from your parents, but you get 2 copies, right? One from your mom and one for your dad. So that's why there's 2 boxes. Like this box, we could basically put an X in it. Let me see if I don't remember how to do that, but let's just make it black so we don't put anything in. There we go. So I know some people like standardize this, like where it's like the females on the top and the males on the side and then the offspring are in the middle. We're not going to adhere to that. We're not that anal, right? But let's just say, you know, parent 1 at the top and parent 2 at the bottom on the sides. So you get 1 from each parent. So what are the possible, so this your blood type is actually, we would say, your phenotype. But your genotype would be that information on the chromosome that codes for these proteins. Actually, I think they might be protein and sugar, but, you know, again, we're not going to get real particular. For A-type blood, you could be given from both of your parents the information on how to make that protein. Let's just say protein for ease, right? Because that makes sense to us. So you would be AA. And what do we say the term for that is when both your alleles are the same? Homozygous. You're homozygous. Could you be heterozygous? Yes. And how would we write that? Nope. AO. And that's because O doesn't code for anything, right? But as long as you get 1 A, you're going to be A, right? Still with me? So same thing for B, right? You're either BB, got 2 B's from your parents, or you're BO, you know, not BO, but BO, right? Which basically O was meant to be a 0, y'all. It was meant to be nothing. But you know, O's and zeros can be confused, and they really just didn't designate, and so since they had letters, everyone kind of stuck with O. So that's a little backstory on that. Okay, so if you're AB, that's it, right? That's your genotype as well. You got an A from one parent and a B from another. If you're O, there's also only one possibility. You have to be OO, homozygous. Everyone still with me? Okay, so I'm going to backtrack. I'm going to put myself in this Punnett square, right? I'm going to be the offspring. So I am O, so I have to be OO. I'm homozygous, right? I don't produce any of those proteins on the surface of my red blood cells. So that means I got each one of those O's from each one of my parents, right? So I can back up, and each one of them has to have an O in order for me to be O. Crap. Formatting's off. O and O. Okay. My dad was O positive, right? So I can put him up here at the top. So he's definitely like me, OO. My mom is A. But we know that she's not homozygous, right? She's heterozygous because she had to have an O to give to me to make me O. Everybody still with me? So what's the possibility for my sisters, who I did not ask? I haven't done all my homework yet, right? So if we fill in the Punnett square, right? So you put all your O's down, right? So everybody got an O, though, from Dad, because that's all Dad had to give was O's. Everybody still with me on that? Mom, we know because of me, could give an O, and Dad could give an O. I should probably just put a caps lock on. There we go. O. So, that's not an O, is it? O, okay. O, why would you put the 0 right above the O on the keyboard? Whoever did that is just evil. Okay. So then we would put the A's in, and typically we would write the dominant one first. So I'm going to put it in front. So that is kind of a standardization I'm going to keep with. So that's dominant, that's the one that's expressed. And again, it isn't necessarily, how do I explain that? We'll come back to it. I'm not going to worry about it for right now. So what could the genotypes and phenotypes of the offspring be for this cross? So for this cross, let's insert another text box over here. So for this family cross, my parents, my family, genotype for the offspring possibilities are AO and O. Yes? So the phenotypes are either A-type blood, right? Or O. Could we have somebody with B blood type in my family? No. Not in my family. Not if you truly are my 100% sister or brother. Which I only have sisters. Okay? Because these are the only possibilities. And this is why at one time, to a certain extent, they could figure out, for some of the crosses, we'll look at one where you can't, you could figure out paternity. Right? So for instance, for, let's finish up this one. So, what's the ratio here, or the percentage? 50-50, right? Or 1-to-1. Does everyone see that? Right? So, and it's for both the genotype and the phenotype, right? So the genotype, actually I could just add it here. Let's do 50%, 50%. Okay. Mm-hmm. No, it is possible. Depends on, how that came about. But it is actually possible. It depends on what type of twins you're talking about, though. That could only happen with fraternal twins. And what fraternal means is they shared the same, what twin means is you shared the same womb at the same time, right? That's what twin means. Identical means that you literally were the same egg, and at some point in the division, you split into two individuals. And that's why they're genetically identical and look alike. And you can't have boy-girl. It's two girls or two boys or whatever. Fraternal means the woman actually released more than one egg, and each one of those was fertilized with a separate sperm. And so that's why they can be different sexes. And technically, the sperm could come from two different guys. It is possible. Yeah, pretty damn close.
That's got to be very rare, though.
You'd be surprised. Mhm. Well, and that's why, no offense, I didn't want to say anything, but gang rape, right? So, yeah, it's possible. It is possible. If there were two eggs in there, or more than one eggs, and a whole lot of spermies. It just depends on who wins that race. Okay, so, let's just say why they don't talk about this kind of stuff in grade school anymore, right? It's even harder at the collegiate level sometimes. Okay, so and then if we were to do ratio-wise, right, both of these, as we said, would be a 1-to-1 ratio for both genotype and phenotype. So we'll, and I'll just write and phenotype. So we'll come back to this story. I'm going to find out what my sisters are, right? They're either A or O, right? So I know they know. And they've told me at one time, but I can't remember, huh?
I know what all my sisters are.
Oh, cool. We'll do yours
Just
next.
had to talk about it.
We'll do yours next. Let's do yours next. Okay. So, do you know what your parents are?
Yeah, my dad's O positive and my mom's A positive.
Okay. Do you have any O's? Do you have any siblings that are O's?
Yeah, 2 of my sisters are O.
Uh-huh. So you're the same Punnett Square as my family.
Yeah, well, and like with the negatives and positives, but 2 of
Right.
us are O.
Yeah, we're not going to do the O's and the we're not going to do the positive and the negative. That's separate.
Okay.
We can do it separate if you guys want, if we have time. Okay. So she is the same Punnett Square as I am, right? And so what is yours?
I'm A. And
You're
my
A.
little sister's A, and then my other sisters are O's.
O's.
So we
Yeah.
got the exact.
Yeah, the exact same Punnett Square. Cool. So you know that who is it A? Which parent is A?
My mom.
Your mom. She's AO. Because there's no way you'd have O children without her being heterozygous. Make sense to you guys? It's kind of cool, huh? All right. So for me, and that'll be it for me here. I'm gonna copy this one.
Does my dad have to be homozygous?
Yes, O's have to be homozygous because it's a recessive trait. Yes, you have to be OO, and that's all you can give is O's. So the formatting was off on that one, so oops, I screwed that up. All right, here. All right, that should be right. Yeah, really, Erica? There we go. That should give us enough to play with. Okay. Anyone else want to share? Figure out theirs. Okay. Both of your parents are A, and do you know what yours is?
A.
You're A. Do you have any siblings that you know?
Yeah, they're A.
They're A. Okay. So, what do you guys think about that? If she's A, right, both her parents are A, so we can put an A at least, right, in these squares and come up with this. But then, do we know if this is O, right, if they're heterozygous? How many siblings do you have? It's you and okay, so there's two of you. So, yeah, but within these two parents. Okay, we'll stick to these two parents. So considering that there isn't any O's, but does that mean that there couldn't be? No, right? Because, I mean, it's a 50-50 shot, and if you've only got two siblings, you know, probably everybody is homozygous, right? I would say probably. But we don't have enough data to know that this truly is what it is. Because it could be, right, our other possibility for those phenotypes, because that's all we know is phenotype, right? That it could be this. The only way we would know for sure is obviously to do testing, or if we had, I did this wrong. This happens when you try to work backwards. AO, AA, and AO. So for this one, this is the other possibility with A. Well, actually, we could do a third possibility, right? Yeah. So they could both be heterozygous, right? So you could, and you would know this especially if you ended up with an O child, because that means that both parents are heterozygous because you ended up with an O. And so both parents had to each give an O. The other one, if you think about it, and you could flip this one either way. I'm running out of space. Is if one was heterozygous and one was homozygous, right? So one is giving nothing but A's, where the other one gives an A or an O. Would you be able to know this by looking at the offspring? No, even though the genotype is different for this set, phenotype, it's all 100% what? Everybody's going to be an A. It's only if you have two heterozygous individuals, right? And that's what Gregor Mendel looked at, right? Is he took two pure breeding streams and he made heterozygous individuals, and then he let them self-fertilize. So then you got back that recessive trait.
So only if you have two O parents would you actually know? I mean, is that the only way to know the offspring?
Well, if you have two O parents, what do all the children have to be?
They're all going to be O.
They're all going to be O.
So that's the only way you could actually know if that blood type, if it, like one of the parents wasn't the real parent using blood type.
Oh, no. I'll show you. I'll show you. So, okay. So, yeah. Okay, so for instance, yeah, if one of the parents is O, it makes it easier to a certain extent. But, okay, so I'm O, right? There's two Punnett Squares because I don't know my son's blood type yet. I want to find out, though. So, copy. Oh, here, yeah. That works, too, because then I can just change it. Just so that it's... Sometimes I do these ahead of time, but I think it's good to kind of work through it together. Okay, so this is the one of the possibilities. My ex-husband is B, right? So he could be BB, and in that case, our son has to be what? B. You see that? That's the one I'm filling in right now. But if he's BO, our son could be O blood type. Or he could be B. Could he be A? No. Because the A has to come from somewhere, right? So that's definitely when it's like a dead giveaway. Like when one parent is A or B and, you know, he's like, okay, where did that come from? Is anybody this combination? No? Okay. Anyone want to share theirs? There was more hands. Huh?
We're all O's in my family.
You're all O's. Oh, yours is easy.
Yeah.
Makes sense. So there were other hands earlier?
I'm all O's.
You're all O's. Oh, okay. All right. So this is the one where it would be really hard to say this kid is not your kid. Okay. Can anyone think of one combination that we could write here where you get all four possibilities down here? AO and BO. And at one time I thought this is what my family was, but it's not. So I used to say my dad could not claim one of us. All right, where did I mess up? There's AB, BO, AO, and this becomes OO. So there's all four, right? So they could have all the four possibilities. Right? They could have an AB child, they could have a B, an A, and an O. But we would definitely know if you had an O that both parents are heterozygous for the A and B blood types, right? And so would be any offspring they produce that are A or B, they're actually heterozygous.
When you get a blood transfusion, do they check your genotype or
Just
just your phenotype?
your phenotype. Because it really, what matters is what's expressed. And remember, O is no expression. And that's why it's so great, because you don't have anything on red blood cells for antibodies to attack. So yeah, you don't actually know your genotype unless you can do a cross that'll help you figure it out. Right? Like for my mom, we figured out she's got to be heterozygous because I'm O. So let's see. So we did the OA, and we did the A possibilities. We did the BO possibilities. And the same thing for B, right? You could just switch this out for B, right? So let's just for fun do an ABAB. I have my little book out. Just remind me after class that you came late. All right, so let's do an ABAB. So what do we end up with that? So genotype, right? We end up with 3 different genotypes, right? So we end up with 1 AA, 2 ABs, and 1 BB. But phenotype, so phenotype 2, we end up with 1A, 2 and 1 type B. So these are the type of crosses that I'm going to ask you to do, right, with pea plants. Except the type of crosses we just been doing do have some dominance, right? A and B are both dominant over O, which is producing nothing. Does anybody know what we call this when you have both A and B being expressed? So one isn't being dominant on the other, right? It's codominance, right? In the ABO blood group, it shows codominance. And again, this is a later concept that I kind of introduced early, mostly because I wanted to do something we could figure out, and like I said, we could relate to. So we'll start now going a little bit more into, how we write the different things in the Punnett squares. But everybody knows how to fill one in, right? And we've done a little bit of practice with that now. All right, you put the parents at the top, the individual alleles that would be in the sex cells. Each parent can only give one. You just bring them down, and the offspring get 2, right? 1 from each parent.
So, like, the 2 at the top, is that, like, mom, and then the 2 on the side are dad?
That's what I'm saying. You can do it. I know that some people, like, keep to that system, and I can't remember what it is. I don't keep to that system.
So it doesn't really matter.
Yeah. I mean, like, especially when you're dealing with, yeah. I am not going to hold you to that. But 1 parent at the top and 1 parent on the side, and in the middle is the offspring. Right. So,
So, like, how does it happen that, like, if your dad has brown eyes and your mom has blue eyes, that you get green eyes?
and that's why I don't want to do that one, because it isn't as simple as just 1 gene.
Okay.
It's several genes coming together to determine that color. Right? Isn't just 1 thing. Where blood typing is definitely just 1 thing. But in the case of the ABO blood group, you've got codominance, and you've got multiple alleles. So let's actually talk about RH, which is the plus or the minus, right? So that's a different blood antigen that we talk about. So this will actually be a good one.
I have a question.
Mm-hmm.
You know how they say, like, having twins runs in the family?
Mm-hmm.
Like, and it might skip a generation.
Mm-hmm.
So is that something like genetic?
Yeah, they obviously believe there's a genetic link and the ability to have twins, both fraternal and identical. Okay, so with plus and minus, this is an antigen again called the RH antigen, or sometimes the D, and you either have it or you don't. So if you're positive, right, you have it. So we denote that with a plus sign or a minus if you don't. But if you have it, are you homozygous or heterozygous? You don't know, right? Unless somehow you end up with a child that doesn't have it. Okay? So for instance, if you are heterozygous, right, and you mate with someone who else is positive, who's heterozygous, then some of your children could be homozygous, right? They'll have it. Others will be heterozygous, like we just saw. Oops, that's not a minus, that's a minus. The minuses don't show up real well. I don't like using minuses. Plus, minus, and minus, minus. I need a space in there. Minus, minus. Right? So again, this is kind of like the OO, right? If you end up with a child that doesn't have the RH, right? They're negative. They're negative because both parents did not give them the ability to make that antigen. So therefore, both parents would have to be heterozygous. So even though both parents phenotypically are positive because they have one allele that's making that one protein on the surface of their red blood cells. Now, both my parents were positive and I'm positive. So it could be this situation, right, where they're heterozygous. Unless we have a negative person in our family, both the parents are probably, yeah, they might be homozygous. So this is where, too, like, why do we want to know? Why do you want to know if you're positive or negative? What's the big deal?
If you're one or the other and your child isn't, it will try to reject the child.
Nope. But close. It does matter for mom, right? Hemolytic disease in the newborn. Who has a problem with that? The mom. What mom? The one who doesn't have the antigen. The mom who's negative, right? If a mom is negative, right, she doesn't have it. If she gets exposed to positive blood, she'll make antibodies against it. The type of antibodies that can cross the placenta, right? So if she's negative, but the problem is, okay, so she's negative, so she's not going to give positive to her baby either, right? So she's negative, definitely. She doesn't produce it. Has to be homozygous negative. If she mates with someone who's positive, right? So even if they're heterozygous positive, right? If they're just got one plus and the other is negative, there is a 50-50 chance, right, that their child will be positive. And that's the problem.
My mom.
Yep. And but let me finish this statement and then I'll answer it. So with this, right, you end up with a positive kid. And then if the mom, if there's some exchange of, or if the mom's been previously exposed to positive blood, she has antibodies that would cross the placenta and attack that baby's red blood cells. Now, if both parents are negative, do you have anything to worry about? No, because it's all minuses, just like it would be all zeros or all O's for O-type blood. Make sense? Okay, where's your question?
I don't know if you see this documentary. It was on TLC when this lady, she had some kind of genetic disorder where she had kids and like it would do the DNA testing on the kids and stuff like that, and it would come back saying it wasn't her. She didn't go through the.
She had two years.
Yeah, she like had 4 kids and one of the kids got sick, so they needed something from both of the parents and stuff, and it was like, oh, she wasn't the mother, but she was the mother and stuff, and she had to go through this little court procedure and all that kind of stuff. It was on TLC. It was up north somewhere.
But any number of things can happen with that. But you would have to know the whole story to be able to kind of, yeah.
There was 2 sperm at the time of inception or something, and I think she developed with one, like part of it somehow got into, she like.
It was complicated.
Yeah, it's a complicated story, but it is.
But there was a 3rd factor.
Yeah, but it was a documentary. It was a live documentary on TLC. I might have to pull it up, but it was crazy.
Crazy things can happen with biology.
Think about it then, like there's 2 babies in the womb or something, and then one of them consumed the other. That wasn't actually.
Oh, so maybe a chimera type thing.
Yeah.
Gotcha. Okay. Melding of things. Okay, so and then too, the other possibility, like if you have a negative mom and the dad is positive, all the babies are going to be positive, right? So I mean, every time they're going to have this problem, potentially. And Dr. LeBlanc, who was leaving out of here, his wife is Rh negative. And they have 3 kids, and I mean, it was murder for her because she had to go through shots throughout all of her pregnancies to keep her from killing her babies. So it was not fun. So the good news, when I was pregnant, I knew I was O, but I didn't know if I was positive or minus for the RH. But I had never been pregnant before. I'd never had a miscarriage. I'd never had surgery. I'd never had a blood transfusion. So it was a pretty good chance that I had never been exposed to positive blood, that I would have made antibodies that would potentially attack my baby. And then come to find out I was positive. And so I had no worries, right? Because you're not going to make antibodies against yourself unless you're experiencing autoimmune disease. But that's where, like, you know, we talked about last time, and I haven't done any research into it. I don't know if any of you guys did on why they did blood testing when you wanted to get married in the past. And I think maybe one of it was to maybe precursor knowing things like this.
They didn't have, like, what is it, ROGAM or something?
Yeah. I don't know if they even... I think ROGAM came about pretty soon after we understood this, though.
It's like the 60s, right?
Yeah. So... but again, now we can, you know, just test the moms. Like, whenever you're pregnant, they're going to, I mean, they come at you with a set of tubes, I swear. Like, the pot is this big, right? You're like, am I going to have any blood left when you're done? I mean, they test for everything, including sexually transmitted diseases, for fear that you may pass it on to your child. So, I mean, and I'm like, I look at the report later, I'm like, I didn't know you were running that. I was like, well, it's good to know I don't have any sexually transmitted diseases. This is good. Right? Like the list was very long when I was pregnant of all the things they tested for. Blood type was one of them, even if you knew it, you know. And I knew mine because I used to work at a school where they let nursing students prick our fingers and test. And they just did the ABO blood group. The antibodies for RH aren't as stable. So they would, you know, we would actually test our own blood. The students would test the instructor's blood at that school. I don't even know if they do that anymore. I mean, everybody's so terrified of bloodborne pathogens, you know. But mine wasn't very exciting because no clumping happens, right? I have no antigens. So it's kind of cool. I was like, yeah, y'all can do mine, but it's not exciting. At least I don't think it is. But, you know. All right. The other cheap way to figure out your blood type, anyone know? Go donate blood. They'll send you a card in the mail. And if you're like, oh, like me, they'll send you promotional stuff all the time to try and get you to go play blood. Yeah. Yeah. I think they call everybody.
Yeah.
My problem is I have really low blood pressure, and I'm almost passed out when I gave blood, so I have to like make sure that I have someone to drive me and that I eat a lot and drink a lot before I give blood. So I haven't done it again since, because it was pretty scary. But they had the van here at school, and I was like, oh, I'm gonna be, you know, I don't have any other classes today, I'm gonna be a good citizen. I kind of regretted that a little bit later, but one of these days, I plan on doing it again. It's complicated for me. So do it if you can, though. It's definitely something worthwhile. So the Punnett Square is, of course, a grid that we've been using that enables one to predict the outcome of simple genetic crosses. And again, you gotta, you know, have the information. So it is actually not Mendel, right? But we use Mendel's work and put it into Punnett Squares. But it was another scientist, a geneticist, Reginald Punnett, so hence why we call them Punnett Squares. They're named after him. So the heterozygous, where both parents have different alleles. We write down the genotypes of both parents. And so in the case for tall plants, we write T to denote dominant and little t for the recessive form of that trait that we're looking at. And we always pick the letter for the dominant trait usually. So in this case, T for tall. So both of them are heterozygous, right? And so each parent is either going to give a dominant capital T or a lowercase recessive t. Those are their gametes. So we have our empty Punnett square, and male, typically, I guess I put at the top. Females go along the side. And we would fill those in like we did, and then we just fill in the rest of the squares. So they shaded that one differently to denote that you would get back that dwarfed there because of the two recessives, one from each parent. So then just as we were doing, right, you'd figure out the ratio of the genotype, which in this case is 1 to 1, or 25%, 50%, 25%, if you wanted to do percentages. And then the phenotypic ratio, though, would be 3 to 1, right, what you actually see. And we know that even though if you're heterozygous, just as long as you have one of those dominant ones, your genotype may be heterozygous, but your phenotype is going to be, and this is wrong. Oh, haha. It's a question. I forgot I got to the questions. And again, guess where I got them? From your book. And so I already said the first one's wrong, right? What should it say? It should say tall, right? Everyone's in agreement with that? Okay. So this phenotype, right? Phenotype. Is that how we write a phenotype? No, that's a genotype, right? So that one's wrong. For this genotype, its phenotype is tall. This is the one that's correct, right? Because again, they switched the wrong terms here. So it's C. So for TT, heterozygous, crossed with homozygous, what is the expected outcome? Homozygous recessive dwarfed, right? What's the outcome? So see if you can draw out that Punnett square real quick and figure it out. And of course we're looking at phenotype here, right? Because they're writing tall and dwarf. You got it already? Okay. We'll see. It's C?
Yes.
B.
Oh boy. That's the Punnett squares. Yours look like that? Okay, how many of them are tall? The two TT heterozygous, right?
First one, 'cause
What are
somebody didn't
these?
say too dwarf or too tall. They didn't say that on the question.
No, but 2 to 2 would be the same thing as 1 to 1 in ratios. You always want it in lowest terms. So remember, I'll rewind.
So how is it possible for two dwarfs to have average size kids? Like, all
Well, again, I don't know how many genes are involved in that, for one.
okay.
And two, remember, each parent has 2 sets, and depending which set, right? So, you know, they could be both heterozygous for dwarfism, say. So there is the possibility they can have tall children or dwarf children. So looking back at this first one when we wrote these ratios, y'all. So notice, again, this was 50-50, right? Or if you counted 2 to 2, but what did I write? 1 to 1. Right, you always want to keep it in lowest terms. And I do that, of course, because I've been doing this for so long without even thinking about doing that. So where are we? This one. Okay. Yeah, so the answer is B. And there's the Punnett square for that. So I am thinking, well, we have 15 minutes, huh? Yeah, keep going. Okay. So the next is the law of independent assortment. And like I said, he really got kind of lucky in that what he looked at were on different chromosomes. So this is when you look at more than one thing at the same time, right? So this is where you're looking at tall or, tall versus purple or white flowers. And like I said, they ended up being on different chromosomes. So he was able to demonstrate this law that, they segregate independently of each other. Okay. So that's kind of like how I was saying before, you have one complete set of chromosomes you got from your mom and one complete set you got from your dad. What you give to your children is a mixture, right? You don't give all the stuff you got from your dad to your kids, right? You give a mix of what you got from your mom and dad. And this happens independently and randomly. And this is why things can get pretty mixed up and you can end up with these big differences where, children in the same family look very different from each other. I still haven't found a good picture of me and my sisters to show you guys. But one of them definitely does not look like the other two of us. She's dramatically different. But if you really look, I'm sure there's some similarities there. So we need to be able to analyze experiments involving two-factor crosses, define the law of independent assortment, and that's why I said where they separate independent of each other. Okay, and that's if they're on different chromosomes. If it's on the same chromosome, you can't do that, right? Because we don't split up our chromosomes, but each chromosome separates independently of each other. So I'll be able to predict the outcome of two-factor crosses. This is when the Punnett squares get really big and you start losing your mind, right? And we can actually do three-way crosses, but usually people don't do squares anymore because, again, that would be too big. And so we do the trees. We draw the decision trees. So we might do one or two of those, but I don't want to spend too much time on that. I'd rather talk about other stuff. I don't want to spend our whole semester doing Punnett squares and decision trees. What's a loss of function allele? And explain why such alleles are useful to study. Well, we've already kind of seen this, right, in our blood typing example. Right? Loss of function means you don't do something, right? And so in the case of the ABO blood group, the O group doesn't produce something on the red blood cells, right? Doesn't produce A, doesn't produce O. And this really helped us figure out what about my mom? That she's heterozygous, right? So this not being able to do something, again, comes from both of your parents in that case. So we're able to figure out that, oh, well, mom's not homozygous for A blood type, she's heterozygous. So dihybrid, di, of course, meaning two crosses. Looking at two different examples. So say round versus wrinkled seed texture or yellow versus green. So when they take the two pure breeding strains, right, and they produce the heterozygous first generation for both traits. You can hypothesize, right, that you could have this outcome of gametes or this outcome. Well, we know it's this second one, right? Because the R's and the Y's can separate from each other. And again, he got lucky. These traits that he looked at were on separate chromosomes. That's why they're separating like they are. Had they been on the same chromosome, that wouldn't have been the case. Right? If they're on the same chromosome, it would be something like this, right? Where this parent could only give capital R and this parent could only give capital R, capital Y together, and little r and little y together. But we know that's not true. These are actually on separate chromosomes, so these can separate from each other. So then you end up with the possibility, right, that they either give an R and a capital Y, capital R, capital Y, two dominants, or capital R could combine with little y, right? Or little r with big Y, or little r with little y. So you end up with these 4 different possibilities for gametes. Looking at these 2 different traits where you're heterozygous, where you have a dominant and you have a recessive for each one of those traits. So then when you draw out the Punnett square, right, because you have 4 possibilities for gametes on both sides, you end up with 16 squares. And again, you end up with this recessive possibility. You don't end up with a lot of them, right? Because it's a 1 in 16 chance that you're going to get this one, that it's completely recessive. It's wrinkled and green. And so the cool thing about this little diagram too is they put the genotype, right? They filled in the genotype, but then they put the picture of the phenotype. Kind of cool. But again, R is standing for round, right? And that's dominant. And that's why it's the capital R. And yellow color is also dominant, so that's why it's a capital Y. Where wrinkled ends up being a little r because it's recessive to round. And then green color ends up being denoted in the genotype as a little y because yellow is dominant over green. Right? That's why they keep the letter of the dominant trait. And then the recessive one gets written as a lowercase of that dominant. That's the system they use for writing out genotypes. So his data, when he crossed, right, for 2 traits, he ended up with this pretty distinct ratio. When you mathematically, simplest terms this baby down, you end up with this ratio that you've probably seen before. Nine, 3, 3, 1. That's like the magic number in genetics, right? The dihybrid cross for 2 heterozygous individuals, the phenotype ends up being a 9 to 3 to 3 to 1 outcome. All the time. If you look at 2 traits that independently assort or on different chromosomes, you end up with this magic, beautiful ratio. Right, so again, this is actual, but if you were to count out in that Punnett square, and you'll see they have it here in this picture, right? There are 9, there are 3, there are 3, and just 1, recessive. But his actual experiment, right, that's empirical. This is actual, right? His actual experiment. When you do the math, it's pretty darn close to 9, 3, 3, 1. And this is just another way of writing it, right? Kind of the line diagram. I get kind of confused with this one. No, I'm going to teach you guys somewhat of a similar method. It's a little bit easier than this one. This is just a different way of looking at it. This cross, if the parents are recessive, right, for the Punnett square, you wouldn't have to write this 4 times, right? If one of the parents is recessive for both traits, they're only going to give little t's and little y's. So you literally can just write 1 column, which makes it easier too for like paring down your ratio, right? Because we already got this one down to 1. So, and then if the other parent is, notice what? They're heterozygous for both traits. So that's why you end up with the 4 different possibilities. So then for the offspring too, you can end up with 4 different phenotypes still. And it's going to be, you know, a 1 in 4 chance of any one of these. 25% chance for each one of these phenotypes based on these parents. So 1 to 1 to 1 to 1. So let's see. And this is just the dihybrid 9331, just written out on a full slide. I don't know why I do that. Could you do 3 or more? Independently assorting. Yes, but the Punnett Square gets like enormous, right? So I typically like the fork line method, right? And I'll show you guys a demonstration of that next time. And I'll probably just have you do a couple of them for homework. End of the chapter. Yeah, we're going to end there for today. So for homework, I'm going to point it out to you guys. I can't remember if in Connect, but I'm going to go and figure it out. But remember I showed you guys in your book. Let's see. Everybody should have access to the ebook at this point, right? Because this is nice, because in your book, for chapter 2, yep, I'm on chapter 2. Except I want to go to the next section, I guess. We actually get to the Punnett Squares. Alright, so monohybrid, cross, tall versus short. If you click on this, right, it's actually an interactive. I showed you guys this before, right? So I'm going to give you homework like this, but I think I can actually assign this and connect to where you do this. But where you can drag them in, and then you do the Punnett Square, and then you can put in the answers, and it will tell you if you got it right or wrong. Or you can just click help, and it'll give you the answer. Which, if you do that, y'all, and you don't figure these things out on your own, you're going to get it wrong on the test. So try and figure it out, but then check your work. So they have it for tall, they have wrinkled and round, and then they have the dihybrid cross even, where you can change up different ones. So I'm going to post an assignment for you guys. I think I can do it through Connect. If I don't, I'll post it in Canvas for you, and you can use these as aids to help you make sure you get them right. Okay. But other than me showing you fork line, this is going to be it for Punnett Squares, y'all. So those of you guys that aren't huge fans, this is it. I'm not spending an enormous amount of time on these things. All right.